Righty. Thank you for having me. I'm happy today to talk about a lot of the background behind Alzheimer's disease in particular and kind of explore the underpinning pieces underneath the condition itself. This is kind of the bread and butter of what I do in the clinic, so I'm happy to share some of that information as well as do a fun dive into some of the topics and some of the parts of Alzheimer's disease that we don't really get to talk about, or rather that don't get as much spotlight just yet anyway. As mentioned, we'll have some Q&A, but I'm always happy to answer questions in the chat. Might not get to them right away, but at the very least, we'll get to them towards the end, but feel free to ask them along the way. All right, first off, I wanted to give a little bit of grounding because while I'm going to be exploring the concept of Alzheimer's disease and its pathologies, we probably should talk a little bit about Alzheimer's disease as a concept. Recently, there has been some significant discussion within the neurobehavioral field as to what should we call Alzheimer's disease. In particular, the kind of flashpoint for that conversation has been this article from Jack et al. In 2024 from the Alzheimer's Association work group, really working to redefine Alzheimer's disease as a pathological process and giving it that really pathological definition. Now, the reason why this conversation is really starting at this time is because there's been a few very important changes. As mentioned, monoclonal antibody treatment for Alzheimer's disease is now a thing. That's new. We have disease-modifying therapy, and that has never been the case before. Second, there have emerged, mostly one, there has emerged a blood-based biomarker through a number of different platforms for Alzheimer's disease in the form of phospho-tau 217. That's whether or not we can consider that definitive of Alzheimer's disease has been an important question. And lastly, imaging, CSF, and blood-based biomarkers are being considered interchangeable within the ATN framework of defining Alzheimer's disease. I'll get into what the ATN framework is, but all that to say, there's been a number of ways in which physicians have diagnosed Alzheimer's disease over the years. Classically, Alzheimer's is a diagnosis is a clinical diagnosis. You hear the story, you do some testing, you identify signs of cognitive decline and a rate and a pattern that is consistent with Alzheimer's, and you can make the diagnosis. Now, with those core disease therapies and with these biomarkers, whether radiological or serum or CSF, it's become a lot more important to be able to define Alzheimer's based on its pathology. Now, I mentioned the ATN framework, and that stands for amyloid, tau, and neuronal dysfunction. Amyloid beta amyloid being the most strongly associated protein with Alzheimer's disease. We're fairly familiar with it. More relevantly, it is considered the start to the amyloid cascade. There are several markers now that are being used to define a positive A category. This would include some more standard ones, such as A beta 42, a ratio of 42 to 40. Radiologically, amyloid PET is used to describe it. And there are actually several markers of tau protein. There are several measurements of tau protein varieties that are actually measures of amyloid. I mentioned before the one serum test we have that is more definitive now, that's newer. That is the phospho-tau 217. That is actually a marker of amyloid. While it is a tau protein, that particular variation of it and its concentration in serum is more associated with the amyloid phase of Alzheimer's disease than with the development of tau tangles in the disease. The T in the framework obviously is tau. there are markers for tau, but we don't actually have easy access to them. Tau PET does exist, with fluorotau being the marker that's used for it, but it's really only restricted to research at present. There are several other forms of tau. MTBR, or microtubule binding region tau 243, in particular, is a really promising measure of actual tau tangles in the brain. It correlates well with those tau PETs, but there isn't really a very stable or sufficiently accurate platform for measuring that just yet. Lastly, there are the markers of basically neurological disease more generally. NFL stands for neurofilament light chain, GFAP for glial filament proteins. Aside from this is also where you get your general MRIs or FDG PETs to measure either the generalized atrophy of the brain or the slowed function of the brain. All of this to say, this was the proposed framework for defining Alzheimer's disease. A process involving amyloid, tau, and then some supplementary markers. And then they mentioned a few others, including alpha-synuclein and vascular brain injury, just to note, like, these are things that sort of pull away from or would point away from a diagnosis of pure Alzheimer's disease, but are relevant for the profile of someone with Alzheimer's. Right. So again, this is the classic definition of what the pathology of Alzheimer's entails: the development of amyloid, the development of tau, both of these being affected by your genetic risk factors, and these leading to the actual condition of dementia. We'll get into this in a little bit more detail in a bit, because there's a little more to it. We're going to come back to this slide in a little while. But these are basically three different variations on trying to represent the process of Alzheimer's pathology and its development. In general, the core concept remains the same. This is a proteinopathy at the end of the day, leading to neuronal dysfunction, network dysregulation, and ultimately neuronal death. However, our understanding of how we get there has evolved a lot over the years. I'll primarily be talking about the proteinopathy elements of it today, but I am going to touch on several of the other components. Whenever someone asks me about what exactly is Alzheimer's disease, I typically end up, with the patient, I typically will stop at that earlier phase. This is a proteinopathy. It leads to cellular damage. It leads to network damage. And the proteins that are relevant are amyloid and tau. Those are the things that the average patient is going to hear. As physicians, we get to know a little bit more about it, and that more about it is very relevant in the realm of exploring therapies, because while the only treatment we have right now is an anti-amyloid therapy, there are other avenues that we really need to be considering in terms of treatment. Not that they're available just yet, but all hopes for the near future. All right. So this is one of the more classic curves. As you can see, the purple line represents, does this one not actually? Yeah, okay, good. It is on there. Okay, so the first line you see is the measurement of amyloid. So first comes amyloid. It's recognizable in the CSF. Then shortly afterwards, it becomes recognizable on an amyloid PET. After the amyloid PET becomes positive, the CSF tau becomes positive, as in theory does the tau PET. Only after the tau markers are positive, we might get actual findings on an FDG PET or MRI. relevant piece here. While we primarily end up talking about Alzheimer's when we talk about amyloid, while we most often talk about amyloid when we're talking about Alzheimer's disease, the amyloid protein itself is not actually correlated with symptoms. It's not until we get to the distribution of tau protein that we actually start to get dysfunction in Alzheimer's disease. And in fact, if you are looking at a amyloid PET scan, correlating locations on that PET scan with symptoms is not possible. However, doing the same on a tau PET is. So again, this is the classical amyloid cascade model. As is this. So this is the big picture of what Alzheimer's is, the whole process of Alzheimer's entails. There's actually a few more things that aren't on this chart, but it covers most of the pieces, and we'll go step by step. This particular slide is kind of one of my favorites just because it covers a lot of ground. It also talks about about the different therapies that have been tried, a couple of which have actually succeeded, and we'll get into that. Again, first we're going to start off with the production of amyloid beta. We have this gamma secretase complex, which cleaves amyloid precursor protein to generate amyloid beta peptides. These peptides, in turn, aggregate through a nucleation-dependent process. They inevitably misfold. They go from monomers to oligomers to protofibrils. Along the way, they become targets for several monoclonal antibodies, and since apparently that was promised, I will note the protofibril time point in the cascade is when the lecanemab antibody would start locking onto them. And these fibrils, beyond just gathering together to get modified while they're in the CSF space, there's in a few important ways. First off, the N-terminal can be truncated with the reduction of a few different amino acids. This leads to reduced solubility, increases the rate of this oligomerization, and decreases its ability to get cleared. Pyroagglutination takes place, which increases resistance to aminopeptide degradation. This happens a lot more actually once it reaches the plaque stage as opposed to the protofibril stage. Phosphorylation takes place. All the serine peptides become phosphorylated. This is associated heavily with more symptomatic AD. And lastly, that you might end up with isomerization or racemization of aspartate residues. These are normal. That particular change is considered more normal, but it again is just another protein structure change that will lead to aggregation. Once you have all these modifications in place, though, importantly, this collection of proteins that has been misfolded spreads in a very prion-like fashion. Misfolded proteins encounter other less misfolded proteins. They match in conformation, and it spreads throughout the brain. That's the amyloid side of this story. Now jumping to tau. So tau is associated with your microtubules and is a key piece of the skeletal structure. It gets encoded in a number of ways, 6 different isoforms, some of which are more likely to go down this aggregation pathway than others. It plays as mentioned, it plays an important role stabilizing the microtubules and allowing for transport along axons, which, as you might imagine, is extremely key for the delivery of neurotransmitters by neurons. Normally, they have some level of phosphorylation. they are they end up hyperphosphorylated in the process of Alzheimer's disease, which leads to uh, aggregation. Like amyloid, you end up with the formation of oligomers, which at that point they're already capable of seeding further aggregation. Uh, you end up with paired helical figures. You end up with paired helical filaments, which I think this is this is supposed to represent. Uh, and you end up from there. You end up with developing mature fibrils, and then beta sheet rich cross linked tangles. So these things fold in on themselves and again start to spread throughout the brain in spread within the cell, then throughout the brain in a prion-like fashion. This occupies a lot of the tau protein that your neurons are supposed to be using to help facilitate neural transport, and otherwise start to cause cellular dysfunction. Oh, that's backwards. Okay, here we go. So what we're looking at here are the illustration of amyloid and tau spread. There's a little bit of a complication in that story of this all being just amyloid leads to tau, leads to Alzheimer's disease. While that is true, there are a couple of things, there's a little bit of complication there. First off, the location in which these proteins start and spread are different from one another. On the left, you see the Thal stages, the preclinical and then clinical stages of amyloid deposition. Amyloid starts more cortically, and then only later does it move towards the midbrain and lands in that the lower brain and entorhinal cortex. As you can see, in most cases, as a fun thing, in most of these cases, you can see that the cerebellum is pretty free of amyloid. When we do an amyloid PET scan, we actually use the cerebellum as the 0 point to compare it to the rest of the brain too. On the flip side, on the tau PET, it actually goes in almost an opposite direction. It starts in the entorhinal cortex. This is the Brock staging of tau. It starts in the entorhinal cortex, then moves into the limbic system, and then only after that does it spread throughout the cortex. And you will start to be getting symptoms, and you get more symptoms, as I said, as tau itself spreads. So a lot of the important symptoms you're looking for really are happening after tau reaches its cortical stage. That said, I will also note that there are a few proposed how these two relate to each other has a few proposed mechanisms. First, there is actually cross-seeding of the two. If misfolded amyloid, if properly folded tau proteins are exposed to misfolded amyloid, it can actually induce tau fibrillization. Similarly, in relation to the concept of spread, as amyloid spreads throughout the brain, it does still destabilize the local networks, which is thought to facilitate the spread of the misfolded tau proteins. And in both cases, while amyloid is not by itself able to cause neurotoxicity, amyloid in the presence of tau can actually be neurotoxic. So they get to work synergistically in a really inconvenient way. Yeah, this ended up getting slide. Okay. Now, important piece here. This is the first of several important pieces, actually. There are, as mentioned, there is more to this process, to the amyloid disease process than amyloid and tau. The first piece I'd like to talk about is actually the mitochondria. The mitochondria is super relevant to any part of the body where you have cells that are highly metabolically active. And the brain is kind of the most metabolically active cell like region of cells we have. So they are extremely relevant. There are a number of changes that end up happening to the brain, not, I mean, in general, as you get older, but in particular, as someone develops symptoms from Alzheimer's disease. Several things several things that will tend to take place. First, the ability of the mitochondria to spread and multiply becomes impaired over time. Second, the actual efficiency of the oxidative process begins to go down over time, and the functionality of the electron transport chain becomes impaired. And that happens both in the presence of amyloid and tau proteins. The reduced efficiency of the electron transport chain leads to the production of reactive oxygen species. While these are produced normally, the fact that the mitochondria are not working as well and need to work harder ends up with a greater production of these reactive oxygen species, which innately are going to cause cellular damage. The dysfunction of the mitochondria leads to calcium dyshomeostasis. Since the mitochondria play an important role in the regulation of calcium within the cell, also, not to mention that that calcium homeostasis has a lot to do with the cell's overall stability and acts as a signal marker for triggering apoptosis. All of that leads to say that this mitochondrial dysfunction is actually one of the key pieces that leads to neuronal degradation, and in turn leads to symptoms in Alzheimer's disease. That's not even all of it, really. Let's see here. So aside from the apoptotic signal release, this process also leads this development of cellular damage leads to neuroinflammation. Neuroinflammation is something I'll talk about on another slide, but is another important piece of this. Initially, there's a wave of neuroinflammation that is thought to be helpful in that it helps with clearance of these aberrant proteins, but over time, the overproduction of neuroinflammatory markers leads to further cellular damage. And I guess one more detail I'll probably share about the mitochondria. There are some studies that have suggested that this mitochondrial dysfunction, this lower efficiency, and the reduced availability of ATP, the greater amount of reactive oxygen in the cell, might actually predate the development of amyloid and tau. While amyloid and tau do contribute to the dysfunction of the mitochondria, it's possible that one of the earliest pieces in the overall cascade might be the mitochondria themselves. These days, there is an emphasis on trying to treat Alzheimer's as soon as possible, which means that it might be that as a target, mitochondria might be one of the more important ones to try and hit earlier, if they are in fact upstream of the amyloid cascade as a whole. As our next piece, impaired degradation. There is a lot to be said about the process of protein degradation in Alzheimer's disease. There was a recent centenarian study, and one of the interesting things they found about the healthy those that are healthy and of advanced age is that some of the genes that were associated with that healthy cognition actually had more to do with protein degradation and protein digestion as opposed to management of amyloid or tau or even inflammation. While those were relevant, these are important to the process. So again, taking our step back, if Alzheimer's disease is ultimately a disease about protein misfolding and then protein buildup then it's also a disease of failure to process proteins properly. So within Alzheimer's disease, the proteasome as a whole and lysosomes start to also have dysfunction. In the brains of people with Alzheimer's, there's an increase in tau ubiquitination, which normally should lead to digestion by the proteasome. However, it's been found that the activity of lysosomes and the proteasome are impaired in patients with Alzheimer's. In part, this is thought to be due to the presence of amyloid protein, misfolded amyloid protein, which itself actually can interfere with the formation of the lysosome. But also, there's also a theory that there has been a change to post-translational modifications of proteins that go to creating the environment necessary for endosomal digestion and trafficking. So there is a gene associated with Alzheimer's, one of the more common ones, which is ApoE4. The ApoE gene in general, and the ApoE4 form of that gene. That in particular has been found to impair trafficking of the endolysosome. So actual transferal of protein, the initiation of autophagy, and lysosome function in the brain, especially in the hippocampi, become impaired, which might actually explain why that might explain best the contribution that the ApoE4 gene has to the development and the risk of developing Alzheimer's. All right. The glymphatic system. So this is a fun piece of the puzzle too. As mentioned again, this Alzheimer's is a disease of proteins and protein clearance. The glymphatic system is your brain's waste disposal process. In short, so CSF is produced obviously in the choroid plexus, but CSF fluid is also released through every artery in the brain, and in theory cleared by every vein, especially the draining veins that drain into the larger ventricles. Let's see here. So in theory, it's a simple process. The end feet of astrocytes line the blood vessels. Outside of the blood vessels, you have a flow of fluid. Every time your heart beats, a little bit of pressure is sent along the arterial network. A little bit of fluid is pushed out, and some of it is collected back into the veins. There's also internal process to the brain itself, especially while asleep, the synchronous activity throughout the brain that occurs as part of the normal sleep cycle also creates a sort of pressure that helps push fluid through the glymphatic system as well. This is relevant because this purging of fluid, or this fluid process, should be helping remove metabolic waste and amyloid and tau from the CSF spaces of the brain and interstitial spaces of the brain itself. However, the glymphatic system becomes very dysfunctional in Alzheimer's disease. The thought is, and for a couple of reasons. In part, this becomes dysfunctional as sleep becomes disordered, which again is very common as one gets older. In part, it becomes dysfunctional in theory because of the buildup of amyloid around blood vessels themselves. And of course, in part, the further dysfunction of just the arteries themselves and the capillaries due to changes that occur in the setting of hypertension, hyperlipidemia, and diabetes also contributes to this failure of the glymphatic system. These are a few pathological slides that illustrate what are called Virchow-Robinson spaces, or basically enlarged spaces around the blood vessels. You end up with a ring-like halo around the individual arteries where they where they previously have expanded or lost the ability to be flexible, or where you have this buildup of amyloid protein. This actually does also show up on MRIs. While it's not something that's always recognized, the presence of these enlarged perivascular spaces is actually considered a significant risk factor for the development of Alzheimer's disease. It is certainly something I always look for when reviewing the MRIs of my patients. Okay. I briefly mentioned cerebrovascular disease just now, and I did want to touch on a kind of a broader concept to finish things off. As a whole, a lot of my patients do not just have Alzheimer's disease. In fact, I would say most of my patients in particular do not just have Alzheimer's disease. This chart comes from an article by Gibson et al. In 2025. It describes the co-pathology of Alzheimer's disease and Lewy body disease, but also other co-pathologies in general. The blue on this chart represents pure Alzheimer's disease. The inner ring describes the portion of their cohort, which consisted of a total of 296 people as a whole, and then 107 within the dementia-free cohort. But about a little over a third of their patients in the what might be considered a mild cognitive impairment or cognitively normal group had just Alzheimer's disease. Half of that cohort had Alzheimer's disease and cerebrovascular disease. A smaller percentage had Lewy body disease. A larger percentage than that had Lewy body and Alzheimer's and cerebrovascular disease. These are not uncommon conditions. It is, given that the largest risk factor for development of Alzheimer's disease is getting older, and co-pathology gets more and more common as one gets older, more often than not, a patient will have multiple pathologies contributing to things. Earlier, I brought up the ATN framework, and that ATN framework has a category for basically other forms of biomarkers, things like alpha-synuclein for Lewy body disease, or TDP-43 for late, which is a limbic-associated encephalopathy, which has features similar to Alzheimer's, but is not related to this amyloid or tau process, and instead has its own protein pathway. But again, co-pathology becomes the norm rather than the exception as one gets older, and in general, as one develops disease. All right. I think, yeah, that brings us back around. Okay, that brings us back around to there we go. That brings us back around to this chart. There are many things that contribute to the development of dementia in Alzheimer's. There are multiple pathways that are contributing to the development of Alzheimer's disease, multiple proteins that are malfunctioning, multiple forms of dysfunction that lead to cell death. It is a vast oversimplification to say that amyloid leads to tau, which leads to dementia. there are, given that there are these other systems at play in the development of Alzheimer's disease, it becomes more understandable that a lot of our treatments to this point have failed. At present, there are two successful therapies. They are both monoclonal antibodies against amyloid. Lecanemab, which targets amyloid fibrils, and donanemab, which targets amyloid plaque. Both of them are helpful. They are not the first anti-amyloid therapies we've tried, and in fact, I would, if you go through the literature, it seems like the majority of research time and effort has been spent on amyloid therapies, but they are the first ones to succeed, and that's great. But their effect is incomplete. As mentioned, once the tau phase of disease has kicked off, it can continue somewhat autonomously and continue to spread via prion-like processes. As mentioned, the metabolic factors and your mitochondria are already malfunctioning and dysfunctional before the amyloid even builds up. So again, overall, it's not surprising that our therapies aren't really there yet for Alzheimer's disease. That is why there are a wide spread of targets that are being researched in the literature right now. Amyloid and tau are two of them, but there are therapies that are targeted towards mitochondria, towards cell death, towards trying to generate neurogenesis, towards trying to control inflammation, towards trying to manage oxidative stress. At the moment, there are, let's see, I'm currently involved in clinical trials. Dr. Liao's clinic at HPN is, of course, also involved in clinical trials. The field is currently expanding. And in 5 to 10 years, it would not surprise me if we had more answers than we do right now. It's been a challenging time, but the more we recognize the complexity of this disease that we're dealing with, the more likely we are to actually achieve some success in managing it. With that said, I feel like I covered a good amount today, and I really would love to hear what questions people have. I don't think I saw much in the chat, but feel free to unmute.
Thank you, Dr. Sonson. This is, can you hear me?
Yes, I can.
This is Leslie Maui. So there was an early report that lecanemab might be less effective in women versus men. Do you know of any new data on that or studies?
So I've been trying to keep up to date on that. There's actually, it's definitely true that there's a question of it being less effective in women. Also a question of it being, there's also a question of its efficacy in non-Caucasian populations as well. Neither of those have been thoroughly answered just yet. The data is being run. Right now, the current question, the most recent questions that have been answered about lecanemab have had to do with what we should do with it beyond the 18-month mark. But as for the question of its efficacy in over half of the population is still in question to a degree.
Thank you.
See. The most recent development actually on lecanemab, just while I'm here and while other people are pending questions, is regarding its delivery. Both of lecanemab and donanemab are infusion-based therapies for the most part, but lecanemab has recently received approval for maintenance therapy in the form of subcutaneous injections, which is a plus. Not having to go in to an infusion center once a month is convenient for a number of our patients, and we have a few people started on that now. They're looking into seeing whether they can deliver the actual initial therapy in that format as well. Donanemab has, yeah, still waiting on further developments on the donanemab side. But for donanemab, its most recent development was the alternate infusion protocol, the alternate dosing protocol for the beginning months, which has reduced its adverse effect rate, which is helpful. Any other questions?